We are going to discuss modified Killiani-Fischer synthesis for D-glucose and D-mannose. Why this reaction is called as modified Killiani-Fischer synthesis is that the two scientists, namely Killiani, okay, who proposed the first part of this synthesis in 1886, Killiani showed aldoses can be converted into two isomeric aldonic acids with addition of higher carbon number. And after that, in 1890, Emil Fischer, he reduced those aldonic acid into corresponding aldoses. So these are the two scientists who contributed synthesizing higher aldoses from the lower aldoses. In short, we can say this entire process is about converting a lower aldose into two higher isomeric aldoses. When I say a higher aldose is formed, and we are showing that the two isomers of that are formed, okay, that is obvious because we are adding one new chiral center, and because of that we are getting two isomers. Okay. Now let's quickly see what are these reaction or what is the reaction sequence, okay, which is involved. The examples for these chain lengthening reaction are pentose to hexose. A 5 carbon aldehyde sugar is converted to 6 carbon aldehyde sugar, or a 6 carbon aldehyde sugar like hexose is converted into heptose. Here we are going to discuss conversion of D-Arabinose into D-glucose and D-mannose. D-Arabinose is a 5 carbon sugar, or we can say an aldopentose, which is converted into an aldohexose, two isomers of that, namely D-glucose and D-mannose. There are four reaction steps or four steps involved in this conversion. This is D-Arabinose, an aldopentose, okay, consists of 5 carbon and an aldehyde group. Okay, it first reacts with HCN and gives corresponding cyanohydrine derivative. When this cyanation reaction takes place or the cyanohydrines are formed, as you can see in this picture, there is a new chiral carbon generated, and because of that we are getting two isomers. These are the two structures of isomeric nitriles which form when we do the cyanation reaction. In the next step, these cyanohydrines are hydrolyzed using barium hydroxide, and further on acidification gives corresponding aldonic acids. Okay. Now we said this is Killiani-Fischer synthesis, and as we said, the Killiani who proposed that these aldoses can be converted into corresponding aldonic acids. So up to this we can say the work proposed by Killiani. Okay. Cyanation followed by hydrolysis gives us the aldonic acids. Here we are adding one carbon, so these are having one carbon more as compared to our original monosaccharide. The next step is when these aldonic acids, okay, we can say acids having hydroxyl group form intramolecular lactones. So aldonic acids subjected to evaporation forms these isomeric lactones. Okay. Lactones are cyclic esters. Okay. We get two of these, and finally when these lactones reduce using sodium mercury in presence of dilute hydrochloric acid, we are getting the corresponding aldohexoses, D-glucose and D-mannose. These are the two isomers which have different configuration only at C2 carbon. Here you can see in glucose, the C2 carbon, OH is on the right hand side, whereas the mannose hydroxy group is present on the left hand side. So these are the two aldohexoses, isomeric aldohexoses are formed. So this is the entire sequence for modified Killiani-Fischer synthesis of D-glucose and D-mannose from D-Arabinose.